Dr. Kate Hubbard from Florida Fish and Wildlife Conservation Commission Center for Red Tide Research is going to provide an overview of red tide bloom dynamics. Next slide. The purpose is to provide a briefing on the latest red tide and harmful algal bloom research and present a new suite of tools which can be used to better understand bloom dynamics and increase relevant observations. This relates to the water quality improvement action 5 to reduce harmful algal blooms, specifically activity 5.1 to support HAB research and monitoring and measure to reduce their environmental, social, and economic impacts through ID and reduction of anthropogenic influences. So with this, Dr. Hubbard, I'll turn this over to you for your presentation. Thank you for joining us.
Thank you so much for the opportunity to speak today. Next slide, please. So I wanted to give a little bit of background about Karenia brevis, the red tide organism. This is an organism that pretty much solely blooms in the Gulf of Mexico. Blooms can be transported out of the Gulf to other regions, but it has a long history of blooms here. The duration, location, spatial extent of these blooms varies from year to year. This organism produces a suite of toxins known as brevitoxins. These can cause human health impacts, including neurotoxic shellfish poisoning if they're consumed. They can also cause, the toxins can also cause respiratory irritation when they're aerosolized. And if you look at the map on the top, that's showing the distribution of NSP toxins in seafood over the past 10 years. And again, you can see that's largely restricted to the Gulf of Mexico. There have been a handful of times where we have seen blooms transported around the south part of Florida to the East Coast. There is extensive wildlife mortality during blooms. This is fairly common. It can include fish, marine mammals, birds, sea turtles. These blooms start 10 to 40 miles offshore at depth. I'll go over that a little bit in more detail. And these blooms occur across a wide range of environmental conditions. I think we're all familiar with these blooms, which can last sometimes over a year. And if we think about what that translates to in terms of the environment, that means that it's lasting throughout our different seasonal conditions. And so again, it's a pretty tolerant organism. And you can see it's a single-celled alga. You can see images there and then on the title slide as well. And then you can see some of the high concentrations that we might observe during a particularly severe bloom. These images are from 2018, where we had bloom observations up to 10 miles offshore. Next slide, please. So if we look at the map, these are observations from 1953 to 2019 in FWC's historical HAB database. And it's just showing where we've seen red tide, and what pops out is that there are lots and lots of observations in Southwest Florida. That's because we get a bloom there almost every year. And if we look at some of the other regions, we get a bloom approximately a third of the time in the Panhandle region, less frequently in the Big Bend area. We often have bloom observations in the Florida Keys, and that's partly related to the currents that can move the blooms around. And then, as I mentioned before, we have fewer blooms that have been detected on the east coast of Florida. We think about blooms in different phases, with the initiation happening in late summer and fall. Then we have growth during the bloom, when it typically will peak, but we also have movement around. And then at the end of the bloom, this is something that we're still actively researching, but there may be advection or movement via ocean currents that help take that bloom offshore and disperse it. But there may be other loss factors that are involved as well, such as stress to the actual cells. Next slide, please. It's hypothesized that there's one bloom initiation zone that's offshore. We know that cells are always present in the Gulf, but this initiation zone is located roughly from north of Tampa Bay, the Big Bend region, down to south of Sanibel, and approximately 18 to 74 kilometers offshore. And during late summer, fall, we typically have onshore flow at depth, and that sort of precipitates the sequence of events that starts to move cells onshore via these deep ocean current pathways. Next slide, please. We can visualize this a little bit differently. So now we have the shore on the right-hand side, and we're seeing those cells being moved towards the shore by those deep ocean currents. Next slide, please. As they move into shallower water, we typically do have upwelling. That's the transport of bottom water to the surface, and that's driven by surface winds, which push the surface water offshore. And that process can help those cells accumulate in certain areas, and it also brings them near shore where they're typically observed. And often impacts are some of the first things that are observed. So respiratory irritation, fish kills. We know that this organism can use a diversity of nutrients to grow, and we know that it can use both ocean and land-based nutrients to help fuel that division. Next slide, please. So some of the earliest reports in Florida come from the 1800s, and these reports mentioned there being blooms occurring pretty much every year, and they're interspersed with particularly severe blooms, and that's what we see these days as well. If you look off the coast of Florida, we have a really wide shelf, and the other area that experiences blooms is regular blooms is the Yucatan Peninsula. So you can see there's a similarly wide shelf there. I mentioned our blooms are very frequent, same in the Yucatan. And in Texas, there are blooms as well, but they're transported from the Yucatan and they are less frequent, and they're related to the winds and the currents moving things around along the western shore of the Gulf of Mexico. We do sometimes see those panhandle blooms getting advected west and impacting Alabama, Mississippi, and even more rarely, Louisiana. And then, as I mentioned before, we can sometimes get blooms that are transported from Southwest Florida up to other states, and they've been transported as far as North Carolina. Next slide, please. This is showing some of our longest documented blooms. We've had two fairly recently, unfortunately, that have lasted over 12 months. And then if we look at the longest bloom that has been observed, that was in the mid-90s, 94 to 97, and that bloom lasted for 30 months. This is using a bloom threshold of 100,000 cells per liter. We can also look across the annual cycle to see when blooms have been observed previously. Typically, we have blooms that start to occur in late summer and fall, as you can see in this. That does coincide with the most active part of hurricane or tropical storm season. And so that's really important for moving blooms around, but also helps with some of the unpredictable nature of these events. And we typically see fewer blooms occurring in the summer and spring months. And this past year was an anomaly in that respect. Next slide, please. So just comparing our two most recent severe events, the 2017 to 2019 bloom was the 5th longest on record. It lasted for 16 months. It impacted 22 coastal counties spanning the Panhandle, Southwest Florida, and it was one of those blooms that got moved to the East Coast. The 2021 bloom lasted for 12 months. 17 coastal counties were impacted. They were all on the West Coast. We were experiencing a drought in the spring, and that may have contributed to some of the conditions in Upper Charlotte Harbor and also in Tampa Bay that allowed the bloom to really proliferate and cause impacts in those areas. And these are showing monthly summary slides for both October, during both years, during the most severe part of the bloom. Next slide, please. So I mentioned this before in terms of Karenia being able to use a diversity of nutrients, both ocean sources and land-based sources. This is something that's been widely examined in the lab as well as in the field. In the lab, we can take cells basically out of the field, we can culture those, grow them up, and we can expose them to a number of different conditions to get a better sense for how they may be responding to triggers in the environment. What we know from those experiments is that there are a number of different types of nitrogen that Karenia brevis is able to use to fuel growth. We know that it also has a preference for ammonium, but it can take up nitrate, urea, as well as other forms. And it may be able to modulate that nitrogen uptake to be able to take up different forms. If we look at the graph that I'm showing there, this is a graph showing the different forms of nitrogen located offshore. And the main point of that is just to say that nitrate tends to be fairly low in that environment, and that we have other forms that tend to dominate. We know from some of the work that's been done that there are at least 10 different nutrient sources that can contribute to a bloom. And this may be really important for the different stages of a bloom. We have initiation, sort of the maintenance and the peak, and then termination. There may be different sources playing a role in those different bloom stages. Next slide, please. So this is going back to this slide just to say that with these different bloom stages, we know that Karenia is not just really flexible when it comes to nutrients, but from the lab studies that we've done, and also to some extent from the field work that we've done, we know that Karenia can also tolerate a broad range of light conditions, and it's perfectly happy growing at the high light levels that we have at the surface of the ocean. And this is a species that moves up and down in the water column each day. It moves up to the surface to get light for photosynthesis. It also then moves down at depth at night to avoid predation. And also this allows it to access different nutrient pools even throughout the course of a single day. When we look at temperature and salinity, going back to these observations where we do have these long-lasting blooms, we know that Karenia can occur across a wide range of temperatures and a wide range of salinities. We do see that there is sort of an upper limit for temperature and a lower limit for salinity that seems to kind of constrain its ability to grow. Next slide, please. And so we have currently short-term prediction for Karenia brevis that's focused on ocean currents, but we've also looked at this to try to predict Karenia brevis blooms at seasonal timescales to get a better sense of how severe a particular event may end up being. And looking at 25 years of data and being able to look at particularly the loop current, which is a really important current that occurs out in the Gulf. Looking at the position of that and then also the strength of that, we can try to draw parallels between that and then the subsequent bloom that may or may not occur. And from a study that was conducted by Bob Weisberg's group, Younggang Liu, we found that there are certain patterns of the loop current, again, that are more conducive to setting the stage for Karenia brevis blooms to occur. And you can see that when the loop current starts to impinge upon the dry tortugas, that's when we will often see conditions that are appropriate for pushing those cells onshore. However, there is too much upwelling that can occur, or if things are really, really strong there, what that means is that there's more delivery of subsurface nutrients to the surface, and that likely is in some way inhibiting Karenia brevis from being the dominant type. And going back to 2010, that was a year where that actually was observed, and there was no Karenia brevis bloom observed that year. So every once in a while we do look out and we don't have a bloom in Southwest Florida. As we start to think about the research that goes into predicting blooms, both at seasonal timescales but at lesser timescales, we're continuing to investigate the physiology of this organism as well as the life cycle. There have been some new advances for in terms of the life cycle of the closely related species Karenia mikimotoi that has identified resting stages and sediment. That's something that we're actively looking at. And we have some new technology that we've been deploying in the field that gives us a better sense of when Karenia brevis is actually growing versus when it may be hanging around or stressed. And so this can allow us to get a better sense of how it's actually responding to its environment. And with that, I'll stop and take any questions.
Thank you for that presentation, Dr. Hubbard. Are there any questions?
Yes, Alice White. I do have a question. Could you elaborate a little bit more on that slide you had that showed the blooms in the Panhandle and then the immediate West Coast and then the South. Why that it's so prevalent on the immediate West Coast? And then my second part is, how does that tie in with Mississippi and Alabama not really having the number of blooms that we have in the top portion of Florida?
Yeah, so I'll actually see if we can go back to even an earlier slide that has the pictures of the shelf.
Okay.
Yep. Oops, we went past it. But the wide shelf in Southwest Florida is really important. And that's something that, there we go. That's something that kind of sets the stage for these blooms happening. And with the hypothesized initiation zone that occurs kind of from the Big Bend to Sanibel, we see that the prevailing currents, the direction that they flow in our area, is to bring cells to Southwest Florida. One of the questions that we have about the Panhandle blooms that we're trying to actively look at is whether those are the same populations as what we have in Southwest Florida, so we can use genetic tools to address that. But in theory, if we do look at the prevailing currents in that broad area, at the northern part of the initiation zone, some of those currents may transport cells to the Panhandle region. And one of the questions that we've had, especially with the most recent blooms and thinking about how those developed, we had these long-lasting blooms, and at some point we did see cells in the Panhandle. We don't know if that transport occurred due to tropical storms. There were really big storms that happened both years, especially in 2018. And when those storms happen, we lose our ability to track these blooms from space using satellites because of the cloud cover. And so we don't know if there's transport of the Southwest blooms to the Panhandle, or if there's effectively a new initiation event that occurs there, or possibly a combination of both. If we look at some of those other states, they don't have the right bathymetry to really set the stage for these blooms to occur there. And so we just don't see blooms as often in Alabama or Mississippi. We also know that Karenia brevis does have a salinity tolerance. And so there may be a secondary factor where the lower salinities in that area also contribute to fewer events being able to really take hold in that region. In terms of trying to connect life cycle dynamics, there may be this resting stage in the sediment. It may be that there's simply cells present. And so that's something that we're actively looking at too, to get a better sense on that initiation.
Okay, thank you. So really, pretty much what you're saying is that because of where Southwest Florida sits, it's like the perfect storm. Is that what you're saying? With
Yes.
the shelf, the
shelf
currents?
and the currents.
Yeah. And then also the lower salinity that we have. Is that what you said?
Further north, there's lower salinity typically
further
due to
north.
yeah.
Okay.
So that's something with the Mississippi River input that can lead to lower salinity conditions in the northern Gulf. And so the blooms that we have in the Panhandle typically last 3 to 4 months, and that's it. And so that's very different from what we see in Southwest Florida, where they really are able to take hold and last for a long time.
Okay. So then, just so I understand, so high salinity is good for red tide. Is that what you're saying? Okay.
It can thrive in kind of estuarine salinities and higher. If it gets too fresh, it's not happy. It gets stressed, and then at some point the cells will actually break open due to low salinity.
All right. Thank you very much.
Of course.
This is Mike Miller from Sanibel. What does your research suggest is the ideal water temperature for growth of these cells?
It actually ranges quite a bit, but when we start to see temperatures over 30 degrees Celsius, that's where both in the lab and the field, we start to see that either that growth decreases. We have fewer observations of cells in the field over 30 degrees Celsius. That's not to say that there may not be certain strains that can grow above that temperature, but that does seem to be a cutoff point.
Okay, thanks.
Any other questions?
Yes, Mr. Chairman, Ken Doherty. I'm curious. You look at that zone, that elliptical area out in the Gulf, the initiation zone, Dr. Hubbard. It's really interesting. It almost like it goes dormant out there, and sits out there, maybe in sediment or something, and then something or a combination of environmental factors triggers it. I'm just curious, is that kind of where your research is going, to see if it, what the triggers are out in that area?
Yeah, that's something that I mentioned. There is another species of Karenia mikimotoi, and there's been some really great work that's just starting to be published looking at blooms off the coast of China. And what they're seeing is using genetic tools, they've been able to identify that there are these resting stages in the sediment. And we still don't know fully what the role of those resting stages is and whether or not they're able to effectively seed blooms. And it's something that we're trying to look at in terms of conducting parallel studies for Karenia brevis. When we see at the end of the bloom or during times where we suspect that the cells are being stressed, we see a change in morphology to these sort of rounded cells. And when we look at resting stages for this other Karenia species, we also see these rounded cells. They're a little bit different, but those cells are not going to be able to swim. And so again, trying to put this all together and connect termination of the prior bloom with initiation to the new bloom, it's something that we're definitely working on. But you definitely point out some good things there, where sediment is one area that we'll continue to look in the water column too. And we are looking in the sediment offshore as well as near shore. And we have genetic tools as well as antibody-based tools that we're trying to use to apply to address these questions. And there have been, for other species of dinoflagellates, Karenia brevis is a dinoflagellate, which is a type of alga. They do have almost these seasonal cycles that have been identified. So it makes sense that there may be additional factors and, beyond the ocean currents that are helping these things to start initiate at the same time of year, roughly. So definitely an ongoing source of investigation.
Thank you very much.
Thank you. Is there any last question?
Yeah, if I might just ask, I know you sort of did address this, but has there been a study, you go of 1953 to current day, has there been an overlay on like weather patterns, storms and weather patterns, and is there a correlation to the blooms with weather patterns that we can identify?
Not so much. If we think about the different storms that have happened just in the past 10 years, they all have very different tracks. And
okay.
So there's the potential for those storms to move cells into new areas. There's the potential for them to disperse cells. So there's different outcomes where it could also concentrate cells in a particular area, and that might depend on if the storm's coming from the Gulf of Mexico, if it's coming up the coast of the Atlantic. With the blooms that we see sometimes on the East Coast, the extent to which that may be related to storm systems and the Atlantic, setting up really strong currents that help move things around the south tip of Florida. That would be an example of advection. Hurricane Michael in 2018, that's right around when we really saw the bloom in the Panhandle intensify. So these things are, again, tricky to predict, but it's something that when we have a storm system moving through, we're definitely keeping an eye on what's happening with the bloom.
Thank you.
Dr. Hubbard, thank you again for the presentation.